almost at the end of our list of problems. We got 3 more, 1310, 1312, and 13.A, which I'm just naming the problem we did in class, which isn't from the book. It's well, it's from the chapter, but it's not actually put into a specific problem all its own. So, okay, let's take a look at 1310. 1310 is fun. It is about polynucleotide phosphorylase, and the question says, why doesn't polynucleotide phosphorylase, also known as a CoA's enzyme, which we don't need to know, polynucleotide phosphorylase, why doesn't it synthesize RNA in vivo? Well, why not? So that's not the job of polynucleotide phosphorylase. Polynucleotide phosphorylase, PNP. Now, apparently the job of this enzyme is to take long chains of RNA, so A, U, A, G, U, and I'm using the shortcut method here. So the sticks here are the 5 carbon sugars. These are the phosphodiester linkages. And why not? I'll end this with an OH at the 3' carbon here. And there's a phosphate attached to the 5' carbon here. So here's an RNA molecule floating around in a cell. And the job of polynucleotide phosphorylase is to take phosphates, probably from ATP, I'm not really sure, but what it does is it uses energy to break down this RNA into nucleoside diphosphates. So what we're going to do here is I'm going to be a little PNP enzyme, and I'm going to break off. I'm going to use some energy, probably in the form of ATP. I'm not really sure. We don't need to know that. But what we do need to know is we're going to break this off right here. And we're going to break it off, and when we do that, we're going to add another phosphate onto this. So this whole thing here is going to come off too. This phosphate's coming off. You're coming with me. So we end up with 2 phosphates attached to the 5' end of that U there. A little 3' OH there. Okay, so we have a nucleoside diphosphate. And that PNP, is going to keep breaking down this RNA. It's going to keep, I should probably look this up. It's probably taking ATP, and it's probably using a phosphate from there. I should look this up. But anyways, it's going to keep breaking these down until we have lots of ribonucleoside diphosphates. You can put a ribo here. You can just call them nucleoside diphosphates if you want. It's going to have lots of those, and that's the job of PNP in the cell. But it turns out in biology, when you have a lot of these enzymes, what you can do is you can force them to work in the other direction. And this blows my mind that this can happen. I've never actually studied any enzyme in detail enough to actually understand how or why it works, but I know how it works. And how it works is you add lots of these. You add lots of whatever product the enzyme normally makes. You add lots of that product, and you cause it to work in the other direction. So it normally likes to break down RNA chains, but if you add lots of nucleoside diphosphates, it's going to build them up into a long chain. Really amazing how that happens. But that happens with a lot of biological enzymes, where you put in unnatural levels of one of the substrates or the products, and you can force it to work in the opposite direction. And that's what we do with polynucleotide phosphorylase. So the question is, why doesn't polynucleotide phosphorylase synthesize RNA in vivo? And the reason is these nucleoside diphosphates aren't in high enough concentrations to cause and aren't naturally found in high enough concentrations to allow PNP to work in the opposite direction. Which is probably good, because if it did, it would be making random RNAs, which would be making could be translated into proteins, which could be bad for the cell because the proteins would be meeting this and perhaps toxic. But anyways, the reason it does not synthesize RNA in the cell is because these ribonucleoside diphosphates are not found at high enough concentrations in the cell.